Hello everyone! So today we're going to be talking about sex-linked inheritance. So as you know from previous lessons, you have 22 autosomal chromosomes and one set of sex chromosomes. And so this lesson is all about inheritance that follows those sex chromosomes. And there are a few specific disorders or diseases that do follow sex-linked chromosomes, and we're going to be using those primarily as examples. So, like I said, there are certain traits that appear on the sex chromosomes, which is the 23rd pair. So, depending on your sex, you're going to have different chromosomes. There are X-linked inheritance patterns, which means the allele is carried on the X chromosome. So the trait that we're talking about is coded for on the X chromosome. There's also Y-linked inheritance. So the allele is carried on the Y chromosome. Most sex-linked traits are X-linked. And the reason for that is the X chromosome, well, at least one reason for that could be that the X chromosome is much, much larger than the Y chromosome. So recall from previous lessons, females are going to have 2 copies of an X chromosome, and males are going to have an X and a Y. So right off the bat, we're going to have males and females are going to have different sex chromosomes. So this is where males can sometimes be at a disadvantage when it comes to sex-linked diseases and things like that. Because they only have one copy of the X chromosome, their phenotype is going to be determined by that one copy, whereas females have 2 copies of X chromosomes. So their phenotype is determined by both of those copies. You'll see what I mean in a moment when we get into some examples. So up in the top left corner, you can see chromosomes. And they're labeled. This is the X chromosome, and this is the Y chromosome. And the X chromosome is much larger than the Y chromosome. It holds many, it's larger, and there's more space for different traits or genes to exist on that chromosome. And the X chromosome is going to carry different traits or different genes than the Y chromosome will. So they're totally different chromosomes. So as you can see here, we have the X chromosome, and you can tell it's the X chromosome because it's larger. And we have the Y chromosome, which is smaller. It also says Y chromosome. So all of these words are different genes that exist on the Y chromosome. Don't feel like you have to know them, but they determine different aspects of phenotype, I guess. On the Y chromosome, there's this SRY gene, which is actually pretty important and pretty neat, so maybe we'll talk about that SRY gene a little bit more later in this lesson or in a future lesson. So here are some examples of human X-linked traits. Now, color blindness is an example of an X-linked trait. Hemophilia is another example of an X-linked trait that you may have heard of. And possibly Hunter syndrome, muscular dystrophy. There's tons of X-linked traits. There's no need to really memorize them, but you will see them as we come across certain practice problems. Okay, let's say we're going to do a practice problem for color blindness. Okay, someone just walked in there. What was I saying? So we're doing a practice problem for color blindness. Now, normal vision is dominant over color blindness. Okay, so colorblind. Bear with me as I write this all out. So normal is dominant, colorblind is recessive. So if one thing is dominant, then we are going to give it a capital letter, just the same as you would on Mendelian genetics. So we're going to give that a capital letter, and this is going to get a lowercase letter. Now, the other thing is color blindness is also an X-linked trait. What this means is that it's going to be found on the X chromosome. So when discussing, when writing it down, what you would write is actually XB and XB. And so this tells the person looking at it that it's actually an X-linked trait. So we don't just write the capital letter. You have to do it this way. You have to do it this way where that letter is over top of the X. Now if it's Y-linked, then you would write a Y instead of an X. Now let's say mother is heterozygous. Okay, so, oops, a daisy, that's the wrong pen. Mother is heterozygous for color blindness. That means that she's going to have one big B and one little b. I want you to take a moment and see if you can figure out her phenotype. Is she colorblind or is she going to be normal? Pause the video if you need to. And I'm going to go ahead and write that she is normal. Now we have a special word for individuals who sort of carry that color blindness gene or whatever trait it is we're talking about. So we would call mother a carrier. Because she's normal, but she's still holding on to that color blindness allele, which means she's a carrier and she can pass that allele on to her parents. So that's a special term that you're going to need to remember because you will see it from time to time. Okay, so now let's think of father. Father is going to be colorblind. He's colorblind. Now, when we're writing his genotype, we know that he has an X and a Y. That's his genotype because he has an X chromosome and a Y chromosome. And we also know that he's colorblind, which means he is going to get that little b. So he has that little b for colorblind because he's colorblind. Now, he doesn't have the capital B because he's not normal. He's colorblind, so he has the little b. What I want to do now, what I want to find out is what is the likelihood of these two individuals having a colorblind child? So I'm going to do the same thing I always do, which is set up a Punnett square. What I want you to do is try and place father and mother on the Punnett square and try and fill it out on your own. If you're stuck, then just keep watching the video. But try it out, pause the video, and just check your answers with mine. So I'm going to go ahead and place them. So I'm going to put mom over here. So there's XB and X lowercase b. And I'm going to put dad up here. So there's XB and Y, nothing, because it's not on the Y trait. I'm just going to fill it in. So we have XB, X lowercase b, XB, and Y, X lowercase b, and lowercase b, and here's a lowercase b and a Y. Okay, looking at this Punnett square, these are the possible children. Now, what do you notice about the possible children? I want you to pause the video and just take a moment to sort of figure out the genotypes or phenotypes of the children. What do you notice? Okay, so hopefully you paused the video and gave it a bit of time to think about. Now, one thing you should notice is that we have 50% girls and 50% boys. And you can tell because the girls have that XX chromosome and the guys have the XY. Sorry, female, male. Shouldn't say boys and girls. So that's the first thing you should notice. The other thing you should notice is whether or not they are colorblind. So I want you to go through this Punnett square and identify any that are colorblind. Pause the video if you need to. Now, I'm going to go through and do that. Anyone who is colorblind has all X's with the lowercase b. So that means this individual is colorblind because they have 2 lowercase b's, it's recessive, on their X chromosomes. And this individual is colorblind because he has 1 X chromosome which also has that b. Now, this individual is not colorblind because he's got a capital B, which is dominant and stands for normal. And this individual is not colorblind because, again, she has a capital B, which stands for normal. However, she is a carrier. So there are a few questions that I could have asked you in this problem. I could have said, how many males will be colorblind? And you would say 25%. 25% of males are colorblind. I could say, how many females are colorblind? And you would say 25%. I could say, how many females are, or how many people are carriers? And you can only be a female carrier in this case, so it would be 25%. And I could say, how many males are normal? And you would say 25%. Now, X-linked disorders, as I fix this screen over here, X-linked disorders are more often found in males. So more often found in males. And the reason is that because whenever it's recessive, you only need one of the alleles, you only need one X with that recessive allele to have that phenotype. Whereas in females, females have 2 X chromosomes, so they have an opportunity to get that dominant allele. Here are some examples. We've got color blindness, which is most often red-green color blindness, most prevalent in males. Take a moment to think about it. Do you know anyone who's colorblind? Chances are they're male. Muscular dystrophy is sort of muscles that die. Like the cells of the muscles are going to be dying earlier than they should be. And hemophilia, which is an absence of blood clotting, so that means that they can't form blood clots, which is a problem if they're injured. This image here shows a pedigree. So a pedigree is kind of like a family tree, but for genetics. And what's shown here is different generations. So this is grandfather, grandmother, children of those individuals, and children of their children. And this is a family key showing, actually it looks like it's no particular trait, but it's showing which ones are carrying that trait. So anyone in yellow, let's say it's color blindness because we're using the capital B, lowercase b. So anyone in yellow has color blindness, and anyone in white is either normal or a carrier. And as you can see, males inherit the trait more often than females. Here's another example. This is hemophilia in the English royal family. So Queen Victoria was the Queen of England. Well, she died in 1901, so prior to that. And it was believed that she was a carrier for hemophilia. And basically what happens is you can see most of the people affected by hemophilia are males. And so hemophilia is actually known as the royal disease because it was very common in the royal families of England, Germany, Spain, and I think there was another, though it doesn't come to mind at this moment. And we can use this pedigree to track who may have had it. So our first, these were both normal, so Queen Victoria must have been a carrier, and she passed it on to Leopold. who passed it on to Alice, who was a carrier, and Alice then passed it on to Rupert. And Queen Victoria passed it on to other children who became carriers and passed it on to their sons. Okay, here's another practice problem. Hemophilia is a recessive X-linked trait. So we have two alleles. We have XH, and you can either leave the X blank or you can put a little H on it. So I want you to see if you can create the Punnett square for this trait, or for this problem here. The mother and father are going to be a female carrier and an unaffected male. Pause the video, give it a try. I'm going to work on it over here. Okay. So we have a female carrier and an unaffected male. So the female is going to have two X chromosomes, and the male is going to have an X and a Y. And it says he's unaffected, so I'm going to give him a capital H because hemophilia is recessive. Let me just double check that. Yes. And so the mother is a carrier. Okay, so I have mother and father there, and now all I have to do is fill in my little chart. HH, HY, HY. So the question says, describe the male and female offspring phenotypes. So, can you figure that out on your own? We have one female normal. We have one female carrier. We have one male normal. And we have one male affected. So that's their phenotypes, and of course their genotypes are in the Punnett square. So you can analyze these results a few different ways. You can say that 50% of females are normal, so 50% of females are normal, 50% are carriers. You can say that 50% of males are normal, and 50% are affected. You can say that 25% of the children will be male and normal, 25 will be male affected, 25 will be female normal, 25 will be female carriers. So that's it for this lesson. Make sure to practice this skill, as the best way you're going to be able to do it is through practice. So what you should be able to do is be able to make a Punnett square with sex-linked traits, and be able to tell me the phenotypes, genotypes, percentages of the offspring. But anyhow, as always, if you have any questions, let me know, and good luck.